Yep. Let's sit down here for a minute. I'll be fine in a minute, but it's because I can't swallow.
Great. Well, good evening, everybody. What am I doing here? I'm a former head of Department of Bioengineering, and it's a great pleasure to welcome you and those online, and there are many people online, to this inaugural lecture of Professor Danny O'Hare, our good friend and colleague. In the event of a fire alarm, please follow the emergency exit signs to the assembly point, which is on the corner of Exhibition Road and Imperial College Road. Just follow the crowd. We're not planning anything like that. But in welcoming you, I'd particularly like to welcome, on behalf of the department and the college, Danny's loved ones. Past and present research group members, the Department of Bioengineering family, esteemed members of the college, Danny's broader collaborators. Where else would you want to be spending the evening when Storm Chiron arrives and we can be hunkered down together? And this is going to be a bit of a maelstrom because we're expecting that. It's Danny, aren't we? So just be prepared. Before we start this celebration, I'd like to give a few personal and departmental reflections on Danny's career to date. Danny's been at Imperial on and off for a very long time, since 1981, I think. And he completed his PhD in the forerunner to the department in 1991. And then he secured his first academic position in Brighton. He returned to Imperial in 2001 and has been promoted through the ranks to full professor in 2021, I think. Danny is an academic's academic. He's not an administrator. He has a very strong sense of the priorities in the academic life, what matters. And this is reflected in his focus on, I would say, teaching first, research throughout everything that you do, and service in his own inimitable way. He's a great teacher. Student online evaluation scores are a blunt tool, but he always gets well more than average on that. But more memorable, perhaps, are the comments that they write about him, most of which I can't repeat in public. They reflect on his style, his delivery, his humor, his excellence, and his dedication to them. As an old academic colleague once mentioned, don't focus on getting the students to like you. You focus on learning to like them, and this will help you to teach them and for them to learn well. And I think he does this. And the students simply do love him, and he's always being nominated for Student Academic Choice Awards, etc. Danny has served the department in many ways over the years, in many different ways. But I think all of them take second place to this specific area that he's led significantly, and that is he led the establishment of our intercalated BSc in biomedical engineering for medical students. He has always been a proponent of us, the Department of Bioengineering, contributing to the development of clinical medicine through making them engineers. And the IBSc was born out of this drive, and he led it. He led it from the start. He developed the vision, the syllabus, and got through the paperwork. Strong buy-in from his academic colleagues and from the Faculty of Medicine at Imperial. And he ran it highly effectively, got it off the ground. Since then, he's done some other things, but I just want to highlight that as a key contribution to us here and to all those students. And we're so proud of what we are achieving with the IBSc, and so we should be proud to Danny and grateful to him for his vision, drive, and continued leadership in many ways. Danny's going to talk about his research later, so I won't spend much time on that now, and he's going to bring some new results in as well, actually, so there's going to be some new stuff here that we're all very excited about. But I just want to mention that he's one of bioengineering's strong collaborators. And it's evidenced by his long-running collaborations with many people, with joint publications, joint supervision. These are the hallmarks of a good collaborator. And these are international, as well as UK-based, and within the department as well, and within the college. He also supports enterprise activity, and many of his students have gone on to develop spin-out companies over the years. And before winding up this introduction, and truly, the show is coming, everybody grab hold of your seats, I just want to say he's part of the furniture here in bioengineering. I think there are only three active academics who've been in bioengineering longer than Danny, but none can trace their pedigree back as far as his, if we take into account his PhD. Danny truly commits to the life of the department. Reports of staff-student parties always end with Danny being the last academic standing, or sitting. And we'll all have our memories of other people's inaugural lectures, where at the end of the festivities, Danny stays and stays, like a comfortable old Chesterfield armchair. He is committed to his colleagues and to the vision of the department. He can be relied upon to do the most important things very well, and to support his colleagues and students through thick and thin. Danny, I'm going to hand over to you. Please come to give your inaugural lecture.
Oh, thanks a lot. Thank you so much. Wow, Anthony, thanks for the very kind introduction, and thanks to everyone for coming. What a fabulous turnout. It's almost as many as our first-year classes in thermodynamics, and many, many more than we find in the second and subsequent lectures. So tonight I'm going to be talking about my research and the collaboration. Some of it's going to be an overview of I think, some of the more complete pieces of work that I think we can describe in full. And much of it is going to be incomplete stories and necessary glosses, and I apologize in advance for people whose names I'm going to be leaving out. So, where do I come from? Born, brought up in the English Lake District, where my mother still lives, and my big sister down here in God's own country. Ulverston in the English Lake District, famous for, I think, the saxophone player Jess Gillam, the Nuremberg prosecutor Norman Burkitt, and the navigator Sir John Barrow, but most probably famous for Stan Laurel as his birthplace. In wonderful countryside and gorgeous coastal scenery leading up to these very mountains here that you can see here, the highest mountains in England, the Scafell Massif. I was very fortunate to go to a really good comprehensive school, the only school in town, a proper comprehensive school. And I want to give a special shout out to Miss Wally, who was one of the finest physics teachers in the history of the world, and just was an absolute inspiration, and her love of experimental work. We'd ask her questions to try and catch her out, and she'd say, come back at 4 o'clock and we'll set up an experiment and we'll measure it. This is what you want for a teacher. And Peter Wilde, outstanding chemistry teacher, and we always look back with horror at us washing our glassware with no gloves on in benzene. And amazingly, we're both still alive. Chemistry has this reputation. Even in the early part of the 18th century, from James Gilray and his little friends, right through to more recent cartoons of Nick Kim's, Chemist, this cartoon here called Chemist's Attitudes to Explosions. This very much describes my early years, as my mum and sister will certainly remember. It's an absolute miracle that the house didn't burn down on more than one occasion or be blown to pieces. And Chemist, sinister figure in a white coat who synthesizes chemicals, or a friendly figure in a white coat who dispenses drugs. But what's not to like? Smells, explosions, fireworks, drugs. All human life is there. A chemical industry itself, which still has this reputation as a large and wicked enterprise dedicated to the production and dispersal of pollutants. And I think David Jones's column years and years ago in New Scientist, the Daedalus column, for coming up with these wonderful terminologies. But to be fair, I do work a lot with the medical profession, and it's important to pick your collaborators carefully. Always go for the one on the right. So my career has been totally unplanned. I mean, really unplanned, in a way that I highly recommend to students today. Do what you're interested in at any given moment, and at least you'll have had some fun. If you try and plan it out and think, well, I'll grind through this bit, anything could happen next, and you could miss out on some great opportunities. I read chemistry as an undergraduate at Imperial College, fell under the influence of the legendary Professor John Albury, and having fun was certainly a major part of his approach to doing science, as well as being rigorous about the physics and physical chemistry behind it all. I was keen on research, but I was one of the worst undergraduates in the Department of Chemistry's history. And John said to me, well, I know you want to do research and I won't have you, but there's a couple of interesting, if rather eccentric people over in the PFSU, Peter Winlove and Kim Parker, both of you who are fantastically here tonight, and thanks both for coming. And I fell under their influence. Peter, theoretical physicist, cosmologist, and legendary cabinet maker, and Kim Parker, an actual rocket scientist, who realizes that many of the wave equations also apply to wave propagation in arteries. And I had a wonderful time working with them. It's old enough now that those of you might recognize this were actually drawn by hand in pen and ink on tracing paper, and letrasetted or stenciled letters in my PhD, which covered many of the topics that are still very closely related to what I'm working on now. So maybe not such a random walk. Then did a brief postdoc with Peter before getting a job at what was then Brighton Polytechnic, teaching analysis to environmental chemists, ran into Martin Arundel, whose work I'll be talking about later, great student of mine there, and teaching physical chemistry to the environmental scientists and biomedical scientists there, and teaching physical chemistry to the ungrateful pharmacists. And I was away for 10 years, thought I'd escape the event horizon of Imperial College, but no. Just when you think you're out. 10 years later, I was back. And so this is what I'm going to be talking about. I'm going to, in honor of my mum, I think never miss an opportunity for teaching and learning. I'm going to make you at least try and understand some of the very basics of electrochemistry. There's 2 or 3 people here at least who are going to be absolutely appalled at my summaries. But no matter how much, if I applied all of my knowledge on the subject, they would still be absolutely appalled. So I think a summary is probably what we're going to be doing. I'm going to cover some work on the neurochemistry of aging, and some cells on chips work, and some microfabricated devices before I go on to more recent work. Because I realized when I was halfway through this, it sounded as much like a retirement lecture. And I need to warn everyone, it absolutely isn't. I've got years left of mad ideas I'm going to waste thousands of students' lives on for this. So the last 2 topics that I'm going to be talking about are some work on antimicrobial resistance and our work that we've been doing on that, and on exhale condensed breath, and some work we're doing on translating our work finally into the clinic. So electrochemistry, traditionally thought of as beginning in the late 18th century in Bologna in Italy with the work of Galvani and Volta, originally thought to be a property of animals, interestingly, when Galvani touched his frog leg muscles, they twitched, and it was Volta who later pointed out that it was the corrosion of the steel in the rather than the actual animal electricity. But I think it points to the right direction for electrochemistry, which is at the interface with biology and medicine. But there are older versions of this stuff, like the Smith College Museum Baghdad battery, 250 BC. No one's entirely sure what this was used for. It was used for over 400 years, probably for electroplating, so they could plate gold on lead and pass it off as the real thing. And John Albury again. I mean, it's amazing to see how many people turning out for a lecture with the title, with the word electrochemistry in the title. But there you go. And despite writing the book and being the most eminent electrochemist in the UK for the last 50 years, John himself said, distaste and disgust are the predominant emotions that the normal student feels for electrochemistry. So what is this? And how can it tell us where our molecules are, and most importantly, what on earth they're up to? Loss or gain of electrons. Oxidation, loss of electrons, reduction, gain of electrons. There'll be a quiz after the second drinks reception, so do pay attention. But really, the key idea here is that it's the movement of electricity is the movement of charged particles when you apply a voltage. And these, at the interface between an electrolyte solution and I stand to one side, between electrolyte solution and a conductor, the nature of the charge carriers changes. So here, for example, we've got iron losing a couple of electrons to form iron 2. These electrons traveling to another site in the iron, reacting with oxygen, and the iron itself being further oxidized to create rust. This is happening everywhere on campus as we speak now. But it's also So corrosion is certainly at the heart of it. We can run this process backwards and get electroplating. We can use it to make molecules such as the commercial production of hydrogen peroxide or bleach. And it's also, of course, in batteries. And John Goodenough's lithium-ion battery, which eventually, as I can't believe I'm about to say this, was eventually good enough for a Nobel Prize. But analytical measurements, and that's where I've been coming in. So what do these electrodes look like? Remarkably simple. A piece of metal surrounded in insulator. These are the commercially available devices, maybe 3 or 4 millimeters in diameter, in KLF or some other insulating one. These are some ring disc electrodes. This bar is 100 microns, so these are about the size of the hair on your head. That Sev Harvey, a fabulous PhD student of mine from back in the day, developed a method for building, built an inverted sputter coater to actually make these damn things, and then we measured blood flow and perfusion flow with these. Some microfabricated devices, which I'll be going on to a bit more. Tiny 7-micron diameter carbon fibers, which Martin Arundel here was using in some extraordinarily patient experiments on snail neurochemistry. 7 microns, to give you a scale, is about the diameter of a human red blood cell. And then, of course, the commercial devices for measuring blood glucose. Tony Cass's great invention, the Exactech blood glucose analyzer, which transformed the lives of type 1 diabetics, and all this technology now in wireless form. But the idea here, then, the energy that we have to apply is somehow, through some deep mysterious process, related to the identity of the molecule that's either accepting or donating the electron. So that's how we identify what there is. And how, so voltage is one key to selectivity, but we can couple this to perhaps some other molecular recognition, such as an enzyme reaction or an aptamer reaction. But how fast it happens depends on the concentration. I was told not to put equations in, so out of badness, here one is. The rate at which the reaction happens simply scales with the electric current. So that's how we get the quantity. And this is just Faraday's constant that takes us from electricians' units into molecular scientists' units and back again. So we measure the rate of electron transfer, and therefore we can get the concentration. If we control the voltage, we can control the selectivity, and we can put other molecular recognition events in the way. And that's pretty much all I'm going to be testing you on later on. So it's the only examinable content. So why should we measure chemical concentration? Well, cells only know what's going on their own surfaces. Secondly, we want to measure the concentration because the more crowded they are, the more energy they have and the faster they react. And some concentrations correlate with disease states, such as glucose in your urine, for example, and type 1 diabetes. And so if we can come up with a cheap surrogate for an expensive an extensive medical history taking by an expensive member of the medical profession, then we can be improving outcomes, extending the healthcare and getting good results, and also making doctors' breath nicer after they've just tested a diabetic, because they used to have to taste the urine to do the analysis. The thermodynamics of this, okay, one more learning opportunity. We go from big molecules to small molecules. We get energy in the form of adenosine triphosphate. To get the process to run backwards and assemble molecules, we have to consume the energy. So we go, the biology, from the thermodynamics of the biology, follows this kind of idea. And the general idea that people working in this space are, because there are no real underlying theories, is to try and measure what we can and then gradually piece it together into networks. This last part is a long way from complete. So the general idea is we need some sort of molecular recognition. This might be the voltage, or we might link it to enzymes, like this one from Bhavik Patel here, where we have glucose oxidase competing with hexokinase to allow us to measure and detect ATP. Or we have aptamers, which when they bind their target change shape, and the rate of electron transfer changes. This now needs to be transduced into an electrical signal. So we have different materials. The classic materials are gold, platinum, and glassy carbon. Carbon composites, like we have here. This is a lovely image from some work with Julie McPherson when she set some of our carbon composites up on a fluorescent microscope and did an oxygen reduction reaction. And as the pH rose, the fluorescein, the dye that was in there, the fluorescence was switched on, so we could see the patterns of conductivity and the growth of the diffusion fields. And we also did some nice modeling on this with Hong Zhao, another very talented PhD student. But we can also use fancy materials like boron-doped diamond, and Raf Trouillon is with us tonight and will be able to tell us a little bit about that. Then we need to do some electronics. And I did some stuff with Hunying Lim in Taiwan about this. I came up with some ideas and put these onto a chip for a tiny potentiostat. I must say, I only did this collaboration because it involved molecular imprinted polymers, which I'm a bit skeptical about. But the main reason I did this work was because it was in Taiwan, and it's worth going just for the food. But we need to produce exotic waveforms, measure the current, and process the data, particularly drag the noise out of it, and get improved selectivity by doing some of these fancy signal processing routines that Kostas Anastasiou, who was watching online tonight, developed in my lab, along with some very fancy numerical modeling, along with Chris Bell. Don't read the small print, but the key ideas are here. Why it's so difficult to do any kind of measurement whatsoever in biology. Everything is compartmentalized on every length scale. So if we think of the human population, H. sapiens, every individual is different. You look within that individual, every organ is different. You look within the organ, there are regions within the organ that have got different cell types. Within the cells themselves are compartmentalized. So trying to define what you mean by concentration is by no means straightforward at that point. So that's an interesting challenge. And if you have 1 molecule per cell, it's still picomolar. So we've still got realistic concentrations down at that level. The production of the molecules is very localized. All the timescales are relevant, from sub-millisecond neurochemical signaling up to the chemical changes that happen over aging and the course of a lifetime. The concentrations are low. It says nanomolar here, but really femtomolar and lower can be important as well. The more we improve the sensitivity of our devices, the more we find we need to be able to measure. And we're trying to do this on something that's active, so changing, it's complex, and it's typically moving. And every single part of every single experiment and experimental subject is different. But we nonetheless feel we can make some sort of progress on this. So what kind of stuff has been happening? These collector generator sensors here for measuring flow, generating hydrogen on one electrode, collecting it on the other. This is my own hairy arm here, because you've got to show willing. We're not allowed to do a complete ban on self-experimentation in Imperial College. So I had to go down to the medical school at Southampton. And in return for agreeing to implant my sensor, I had to agree to having my collaborator, I shan't name at this stage, having her dialysis probes implanted in me as well. With some numerical modeling alongside all this, some work on iridium oxide, which we're still doing, boron-doped diamond, Alex Lindsay's photoelectrochemical sensors, where we light things up and look at the redox properties of photoactive species. I even, against my better judgment, did some work on molecular imprinted polymers, signal processing that allows us to separate molecules for which the chemistry would seem different, but we're exploiting the properties of the waveforms to exploit the differences in kinetics between the rates of electron transfer for different molecules. What else? Neurochemistry of aging, which I'm going to be doing quite a bit about in a minute, and these microspike arrays I'll be talking to you about later. But there's some work we did with Alison Willows and Ivan Stoyanov on water quality sensors, because similar problems in sensing apply in the complex wild environment. And I'll be talking a little bit about some new recognition chemistry and some breath sensor stuff later. So into the meat. This animation no longer works, unfortunately, but it was a bit cheesy anyway, so it's no worse really for it not working here. But one of the key things about getting older is you notice how much harder it is to learn. A child learns to ride a bicycle 3 years old in about an hour, maybe less, half an hour. And there's probably nothing more comic than watching an adult learning to ride a bicycle, except possibly watching an adult trying to learn to swim. There must be a chemical basis for this. And one of the aspects of this was to try and measure what's happening in the synapses. So electrical wave comes down this nerve cell here, and it needs to transmit the signal to the next one. And how this works is a bunch of chemicals in these little bubbles here fuse with the cell surface membrane here, burst out. And if we position our little electrode here, we can see a spike in the current as these molecules emerge. And then we see them decay away as they diffuse, or are taken up by the target cell, or are taken up by the cell that releases them. And this is what this device looked like. And Martin Arnold here was involved in the design and making of these electrodes. So serotonin is one of the molecules we went looking for, which can be oxidized. And we get this kind of raw data when the snails were cooperating, which was, I think, what, 1 time in 20? For a 4-hour prep, and we got about 5 minutes of data. It takes a lot of resilience, this field, sometimes. And these tiny signals, picoamp current. And then someone had to go through, someone, I don't mean me, go through and pick all of these little spikes up, zoom in on them and say, well, that's a nice looking one, that's a nice looking one, but that looks like 2 vesicles bursting, or maybe even 3. So we sift the data and then analyze how big they are, how much charge has passed is how many molecules, how high the current was, and then this exponential decay, better than a square root of time decay, showing it wasn't diffusion, but reaction that was causing them to move away, and the reuptake that was dominating. And then when we looked at this, we see the young vesicles were smaller, the middle-aged ones were larger on average, and the old ones were bigger still, so different size and distribution. And this is, we think, a compensation mechanism for the downstream cells being less responsive as we get older. So it's like my son having to shout to get me to hear properly on the phone. You know, it's, so the undergraduates got tiny little vesicles, the postdocs, it's already starting to kick in. By the time you get made professor, you're having to massively increase the concentration of your local drug. And the same thing seems to happen for nitric oxide as well, although the mechanism is different there. It involves changing the enzymes. So next topic, cells on chips. Work from Ralph Truand, Martin Arundel, Tempest van Schaik, and Schurier. This is the early prototype devices, 6 little microelectrodes insulated and microfabricated on a dual inline chip. The more sophisticated version designed by Martin Arundel here, made on glass so we could still see the cells. We apply different voltages. We measure different things. We can measure oxygen at 1 voltage and nitric oxide, NO, a signaling molecule. So if we put glucose oxidase or lactate oxidase enzymes on, we can measure glucose and lactate. If we put iridium oxide on there, we can measure pH, all of the things. So it's kind of a platform technology. And the big breakthrough was trying to find out what makes the cells grow. So coating the surfaces with polystyrene sulfonate polyalysine, which was supposed to be good for cells, turned out to be a bit of a disaster. And you can see the cells curling up and dying. But for B and C on the fibronectin ones, after 24 hours and 48 hours, look at their little faces. They couldn't be happier. So we were able to get biocompatible codes. 2 days worth of measurements is pretty good, and 5 days, I think, was about the record. So here's the protocol. We were interested in angiogenin with the late Suik Chang, a wonderful collaboration that we had going with Suik, starting about 15 years ago, I suppose now. But angiogenesis is where new blood vessels grow. Plainly, that's a good idea if you just cut yourself in wound healing. It's obviously vital in development and growth. But it's a disaster if it's a tumor that's grown to a size that can no longer feed itself by diffusion. It uses those same molecular mechanisms to draw in blood vessels. So an angiogenin is angiogenin here, this peptide, is one of the molecules that is involved in drawing in blood vessels to feed the tumors and provides a pathway for metastatic cells to create secondary tumors throughout the body. And this was Suik's interest here. So we wanted to know, what does angiogenin do? We thought nitric oxide might be involved. So we plate out the 1,000 cells or so on the surface of the sensor array, let them calm down a bit, give them the angiogenin, and we see an increase in the nitric oxide signal. Now, this is numbers and sensors, and the biochemist took some persuading about this, so we needed immunohistochemistry confirmation as well. This is the enzyme that's responsible for this. But we're very confident in these results because there's a dose-response relationship. The more we give it, the more nitric oxide it produced, and we could see the development over time. And a time dose-response curve is generally considered quite compelling data. So we then started looking at the pathways. So neomycin stops the phosphorylation and the AKT pathway and stops it getting into the nucleus. This other drug here blocks the ERK pathway, and this one blocks the PI3 kinase pathway. So we can see the neomycin knocked it back to a control level. This one had no effect, so the ERK pathway wasn't involved. This one, the PI3 kinase pathway absolutely was, because it goes back to control levels when you put this drug in. So we start being able to piece together the mechanisms of how angiogenin is promoting the formation of nitric oxide, signaling the growth of new blood vessels at the very beginning of this process. So what else can we use this for? Well, host-pathogen interactions is pretty cool. This is one of my early forays into microbiology. It involved a collaboration with Di Williamson at DSTL Porton Down, because obviously they're naturally interested in anthrax for many of the right reasons, as well as the obvious wrong reasons, because they do all the vaccine development for this work. It's a very interesting bug. It has a 2-part toxin. One part is protective antigen, and then it needs either edema factor or lethal factor. And it does this extraordinary little dance on the surface. The protective antigen comes in, binds CMG2 receptor on the cell surface. A little bit gets cleaved off. Once that happens, the PA can find 6 of its little friends and create a heptameric pore and drag the lethal factor or edema factor into the cell. But you can work with either protective antigen or either of these two in the open lab, because they need the other part of the toxin to cause problems. So again, the same sort of idea. Exposing it to increasing concentrations at around the 20 microgram level, we start seeing increased nitric oxide production. This time, both the ERK and the PI3 kinase pathway are involved because it's a different enzyme. It's the induced nitric oxide enzyme. Now, we're expecting this because production of nitric oxide is involved in killing any invading microorganisms at the very early onset or recognition of disease. So this is a really important idea. And then by loading the cells with AZT, the anti-HIV drug, we could increase the sensitivity and bring it down to well below the circulating levels you'd find at the very early stages of infection. This is something due to Christian Amator, le grand fromage of electrochemistry in France, who realized that you could actually do this. And we got down to essentially 10 nanograms per milliliter detection on this. So we then looked at engineering different parts of this, work that Di Williamson did at Porton Down. So it turns out this domain 4, which is involved in binding, is all you need. So this part 4 gives the same response as though it was the whole anthrax toxin. So what we were detecting here was the initial binding event, which is absolutely essential for the disease to develop, because mice that are deficient in this CMG2 can't get the full-blown version of anthrax. So we thought we developed a drug screening tool that were for rapid screening for anti-anthrax drugs to prevent infection. And we thought when we braced ourselves for the world to throw money at us, but we often have that moment in academia, right? And then you just wait and there's a kind of uneasy silence. So, brief break. I've always had side hustles in consultancy. And I brought this up because it was kind of interesting. And some lovely work with Howard Dodd, who at least one person in this room remembers. And who was my physichem technician down at Brighton. And I'm only able to mention this because the company's long since got taken over and out of business and no longer in the UK. But the problem was their entire warehouse full of biro refills, upmarket biro refills, dried up. And they came to us, they said, do a quality assurance assay on the solvent that these terrible French crooks have sold us. It's in really poor quality. This must be the problem. Then we can sue them and everyone's going to be happy. We looked at the solvent and it was over spec. Better than it had been sold for. So no problem there at all. So what the hell, what's going on? So we just thought, we sat in the lab, Howard and I, in the afternoon thinking what to do. And so we picked so let's have a look at what the dried up bits on the end of the biros are. So we picked these scabs off the end and ran thin layer chromatography, which almost everyone's done at school, right? With, red cabbage ink and so on. And we've separated the scab out into 3 of the pigments. But then we looked at the ink itself, and there was a missing pigment. Absolutely fascinating. And when we added corrosion products, iron 3 and chrome 3, we were able to reproduce this. So the corrosion was what was causing it, and it was causing precipitation of one of the pigments in the biro ink. So it was electrochemistry after all. But the background to this is kind of interesting. The 6 different grades of stainless steel in a biro refill. I mean, this is insane. And someone really hasn't thought through the supply chain. One of these was a 430 stainless steel, which is only available in one steel yard in the UK. They didn't like being dependent on one steel yard for their supply chain, so they thought, to hell with the consequences, let's spend all the money we can on the highest grade of stainless steel the world knows, which is 316SL medical grade stainless steel, because that's going to be brilliant. And when they told me that, we thought, we better have a look at that. How does it corrode? And of course, it hardly corrodes at all in the normal media. But put it in biro ink, and it was nearly 10 times higher corrosion current density. So we were able to solve that problem, and it turned out to be electrochemistry after all, and not actually a side hustle. And we didn't charge them enough. Why chromatography at that point? Well, chromatography in a more sophisticated form, as HPLC, is really the workhorse technique of analytical chemistry. Now normally coupled with a mass detector on the end. That is pretty much the way everything goes. So here's the column where the separation occurs. You've got a mixture of molecules. You wonder what they are and how many there are of them. You inject it into this flow. They interact in different ways with this column and emerge at different times. And from how much area there is under the curve, you can tell how much there is. And from how long it takes to come out, you can get a reasonable guess as to what they are. And that's the general idea. But we decided to play at trying to miniaturize this technology, surfing on the wave of the craze for lab chip technology in the early 2000s. And with a wonderful student, Pei Ling Liao, built this beautiful little device, but it had, instead of having a detector at the end, it had detectors all the way down. And so we can see at the top There's the mixture all together with one of the molecules. These are neurotransmitters. This is serotonin starting to separate off. And we can see further down the column, the further it's separated from the others. Then this big lump at the beginning starts to separate out into dopamine and its metabolic product, the metabolic product of serotonin and noradrenaline, until finally, halfway down the column, there's full resolution. So we would only ordinarily see this down here, but we were able to use this approach to actually look at developing effective separation methods on a tiny scale with very small sample volumes. But that's not enough. We need to be able to collect the samples. And I'm putting this next slide up for two reasons. Jin Young's in the audience here tonight, and there's also their wonderful animations, or not animations, but actual movies of how do we separate the samples out. Very cunning trick that Jin Young got organized here. Applying oil flow to break up the effluent that comes off the end of the column. So the oil flows in here and here, and depending on what the flow rates are, you can break them into tiny droplets at several hundred per second, or to these lovely wobbly droplets. And each of these is now isolated, and your separated sample is now in a separated single droplets. So some really nice work there, and beautiful engineering. So some more recent work, Xiaotong Meng, and a project here on molecular recognition, which a collaboration with the legendary Sylvain Ledam, 4 rows from the back over on the right here. We both look at each other in a particular way with this, because it's a really cool idea that doesn't work very well. But it's still a cool idea, and we're not giving up. We've already wasted 2 perfectly good PhD students on this, and a postdoc. But there'll be more to come. MicroRNAs are a new-ish class of biomarker, but unlike the others, which are usually proteins, they're nucleic acids, so we can exploit Crick-Watson pairing, the way the nuclear bases line up. We can pair with each other. We can use this for the molecular recognition and get really good recognition on this. So this is DNA with a deoxyribose backbone going down there, and the bases that do the recognition, and of course the chemistry hanging off this. Or RNA has ribose nucleic acid as a backbone, but you can do a synthetic version of this with a protein or peptide called PNA down here, and this is more stable, chemically robust, and has higher binding constant because it's not negatively charged, so there are fewer electrostatic repulsions. And Sylvain has already developed this many years ago into a very reliable assay. So this is the target. You have 2 recognition elements that are brought together by the microRNA that we're trying to measure, which could be a biomarker for cancer, for example. And the one we were interested in with this project is prostate cancer, because it's massively underdiagnosed, and it's typically diagnosed at a late stage. And if we could get a screening tool, we could get better outcomes for the existing tests, particularly if the screening tool could give good results within a minute or 2 on a single visit to a GP surgery, rather than having to rely on men going to the doctor even once, which is often quite a challenge. So there's a serious bit behind this. But this recognition brings these 2 head groups together so they can now have a chemical reaction. And this has been made to work beautifully with fluorescence using a coumarin and a thiol, and you really ought to have a look at this. But we'd like to adapt this for electrochemical detection because it's cheaper, possibly faster, and capable of being miniaturized and distributed and for community use and very rapid screening. So here's the schematic again. The reaction is kind of quite unusual and quite surprising for people who studied organic chemistry, and that was my first love, I think, as an undergraduate, is that you can get this 1,4 addition of a base through this reaction here. But if you use a catechol, this molecule here with the 2 OHs on, this is unreactive. So we can allow the hybridization to occur, then wash the hell out of the sample, clean it all up, wait till we're ready, everything working, then we oxidize the catechol, and the reaction can now take place. So then what happens here, as we can see in this voltammogram, first time through, all we see is a peak for the catechol oxidation. And then when we scan back, the red current emerges, and we see a pair of peaks emerge for this new species that's formed only because the target microRNA biomarker was there in the first place. So we're using the biomarker itself to drive the chemistry. And absolutely, Sylvain needs to get the credit for the templated reaction behind this. And I absolutely take the blame for this working well enough to keep our hopes alive, but not well enough to revolutionize diagnostics yet, I'm afraid. So this brings us on to more recent stuff. I've been trying to get stuff into the clinics a little bit more, and complete devices. The ideas are the easy bit. The implementation of properly engineered technology into biomedical applications or clinical applications is really where the difficulty arises. And I don't know why I'm looking to do something difficult. It's a break with the habit of a lifetime looking for difficult things to do, that's for sure. But the motivation for minimally invasive sensing then is to obviously minimize the damage to the patients and, as an afterthought, the damage to the sensors. But the motivations behind this are primarily demographic. You know, we've got an aging population, as we see here. Look how many people there are over the age of 90. And look at these bulges going up. There are simply not going to be enough people to provide medical care. And the medical needs of the elderly greatly increase. See, four of the 5 graphs here. This is the cost of prescriptions, increasing as we go with age, the cost of primary care generally, community services, which really kicks in on retirement here, a general medicine cost going up here. We see we have a moment of sanity, however, in the mental health here, at about my age, funnily enough. So this is as good as it's going to get. But it's simply not going to be possible to manage the health care of the elderly without getting greater increases in technology. And we get better disease management. We can get longitudinal recording. So, for example, type 1 diabetics' health outcomes are way better now as a result of glucose monitoring. The underlying disease has barely been modified, but their ability to interact with that disease and to manage their lifestyle in such a way as to minimize the long-term consequences has transformed diabetic treatment without the actual diabetes having to be cured or treated. So there's a real role for biomedical engineering in improving lifestyle and outcomes as a result of all this kind of stuff. But there are also applications in well-being, sport, and fitness monitoring where it's a bit of a grey area. But I think in pharmaceutical testing, because it's very rare and very difficult to get more than a dozen people into a phase 2 trial where they test actual drugs on actually sick people. And they're testing them in very artificial circumstances. And we could massively distribute this technology and greatly improve drug testing by doing this. So the 2 areas that I've been looking at, microneedles, and the application here is antimicrobial resistance and also lactate in sepsis, and also exhale condensed breath for exacerbations in chronic obstructive pulmonary disease, and also for diagnosis of sepsis in community settings where it isn't normally possible to take blood. So if we want minimally invasive sensing, where should we go? There's a lovely table from Heichenfeld's paper in Nature about 4 years ago. And the 2 that are in red are the ones you need to pay attention to, because that's what I'm going to be talking about. So traditionally, whole blood, plasma or serum, very well developed. The technology is very well developed for this, because until the advent of scientific medicine, all doctors could do to treat you was draw blood, either with leeches or with lancets. And that's what they did, because that's all they understood about medicine. So technology for taking blood samples was already very good. And since the advent of scientific medicine, the blood samples were there, and we know how to deal with these things. But there's an absolute limit on volume for everybody, but this is a particular problem, for example, in newborn babies or infants, where it can be genuinely limiting. It's painful. You get tissue damage if you have to do repeated measurements. So for the management of chronic disease, this isn't great. And there's a risk of infection if you want to implant something. It's worth mentioning, I think, that continuous measurement really means about 1 sample a minute. We don't need megahertz sampling rates, because about a minute is about how long it takes for your entire blood circulation to go all the way around and back again, through the lungs and back to your heart. So that's how long it takes for the solution to get well mixed. So we don't need to do any better than that. So the alternatives, I guess, that have been used historically, urine, but again, there's variable delay. The concentrations need to be corrected for so you need to measure creatinine at the same time. And so that adds an additional complication. And of course, it's rarely acceptable in public. And so interstitial fluid, which is the space between the cells in your skin, is absolutely available. And you can recover the device afterwards if it caused problems without an additional surgical intervention. And breath, well, what could be, if the patient isn't breathing, then not being able to collect exhale breath is frankly the least of your problems. So as I like to do when we talk about enterprise and tech transfer with students, you've always got to start with the problem you're trying to solve and not the technology, even though in your cold black heart, it's the technology you're really interested in. But this problem, antimicrobial resistance, well, nearly half the population died from infections in the bad old days before antibiotics were around. It's a miserable way to go. It's right up there with metastatic disease. But their resistance has evolved. spontaneous genetic mutation, gene swaps, natural selection, but it's accelerated by excessive, inappropriate uses. And we can see these are the death rates in multiple different regions of the world. And it's maybe 5 million deaths per year worldwide. Premature, 5 million premature deaths per year. That something can be done about. So the effective use of existing drugs is the really key idea here. Obviously, we need new drugs, and there are new drugs like cefiderocol from Shionogi, and there's been about 10 new antibiotics this century. Not many, in other words. But existing and responsible use of the existing drugs means you want the patients to be getting the right dose for the right condition at the right time. And work by Jason Roberts and others, Hagel, show that around 2/3 of the patients in hospital, when they're being treated under probably ideal conditions, are getting the wrong dose. Too high, just right, and too low. And too low is the problem, because that creates an evolutionary selection pressure for the development of resistance. So even under ideal circumstances where they get therapeutic drug monitoring, when they're measuring blood samples, because it takes too long to get the results back, there are still more than 1/3 of the patients getting the wrong dose 5 days in, a dose that's too high or too low. So it barely improves the situation. So what we want is closed-loop control. We want to measure the drug concentration, and that's where our biosensor comes in. Sends an error signal, generates an error signal here. The controller controls an infusion pump, tops the drug up to a target level. Now, this target level could be, for example, a pharmacokinetic or pharmacodynamic level. What's the level that patients traditionally needed for this as a population? But our new idea, and our more recent and exciting ideas, are saying we should be titrating this against host factors. So we should be seeing, is the patient getting better? We should be measuring those factors as well, like C-reactive protein and procalcitonin, because you can stop if they're not getting better after a day, and you can stop the treatment if they get better 4 days into a 7-day treatment. So, microneedles, developed by Tony Cass, look a bit like this, go just into the viable epidermis. So they go through the dead skin at the top of the stratum corneum into the viable epidermis where there are no nerve endings, no blood vessels. So it doesn't hurt, doesn't bleed, there's minimal risk of infection, very small risk of a allergic reaction. So they're very well tolerated. The concentrations of all the important things are very similar to plasma, so we can interpret the values rather well. So we'd like to get the right dose, and our proposed solution is closed-loop control. So how do we get this done? On our microneedles, we put an enzyme that catalyzes this reaction. The red bit's where the action is. So it hydrolyzes that amide, creates an acid. This causes a drop in the pH, which the iridium oxide detects as a voltage change, and we can read this out. And this is what the microneedles look like. And this is, I think, Dave Freeman's arm. Yeah, he's nodding. Patient 3, I think. He was our second legal patient anyway. And you can see after the device comes off, it looks just like the kind of marks you might get on your leg after your socks have been on too tight. So really not, really. And the illumination an hour in obviously has changed because he's now in the pub. But 10 hours later, absolutely no sign. So very well tolerated. And you noticed I went straight in with the safety and tolerance in rather than whether the science was any good. So definitely safe to use. We validated using liquid chromatography mass spec from dialysis samples taken from underneath the skin and venous blood samples taken every 15 minutes. This addresses the essential epistemological problem of analytical chemistry, which is, how do I know I'm right? You measure something, you've got a device you know works, but how do you know you're getting the right result? And validating against techniques that have a different physical principle greatly increases the chances that you're not fooling yourself and not making a fool of yourself in public either when you present these data. So at the top here, we see the 3 sensor curves in blue, green, and red, rising when we swallowed the penicillin dose. The blood levels leading this slightly by about 15 minutes, also oscillating up and down, interestingly, and the microdialysis levels from under the skin, more or less following what we were able to do, measuring just on the surface of the skin. And here's one of our Bland-Altman plots, where we plot the difference between our method and the reference method against the average of our method and the reference method. We'd expect a straight line around 0, and most of the spots fall in this, particularly in the important region where the drug target concentration actually lies. So, Yeah, I've actually managed to keep more or less the time here. I'm more surprised than anybody, and certainly anyone who's ever endured in my undergraduate lectures will be amazed. The final topic then, exhale breath condensate. Our original target work on this was on chronic obstructive pulmonary disease and exacerbations, working with the people at the National Heart and Lung Institute down the road from here. Exhale breath, we've all done it as kids, breathed on a car windscreen or window and seen it steam up and think, well, what's in that? I mean, water, obviously, but stuff that your lungs are exchanging with, that we may be able to find things. It's massively non-invasive. Everyone breathes. We can collect this without hurting anyone, without doing any damage to anyone. But it remains a bit of a controversial subject. The clinical need was early detection and management of exacerbations. In COPD, it's a remorseless disease. Goes on, there's no cure. If there's an exacerbation caused by, for example, air pollution or an infection, they get dramatically worse and stay worse, and they usually have to be temporarily at least hospitalized as a result of that. Now, if we could get early warning from that, the patients could take their emergency test kits, keep out of hospital, have generally more improved quality of life. They could also look back at a longitudinal record if they're testing themselves regularly and find out which lifestyle factors made things worse or better for them as an individual. But there's a lot of controversy about this, and hydrogen peroxide is a pretty good marker, H2O2. But you see here, the lovely graphic produced by Danny Chen, one of my great ex-PhD students here, and control levels are all lower. Within any one study, the difference between control stable, exacerbated, and treated was pretty stable. But you can see that one study here has levels in exacerbation, in stable COPD that are lower than somebody else measured in control subjects. So that can't be right. And the reason for this is that if you condense breath, you sure as hell need to know the temperature of the breath, in and out. You need to know what the flow characteristics are. And because I should have been listening to my own first year lectures on thermodynamics. Temperature and heat mass transport are important in the partition coefficients. The volume that we collect is going to be affected by the temperature. The partitioning of any volatile molecules into the condensate when we collect it is going to be affected by the temperature of the condensate and the temperature of the breath, and we need to correct for that. And Danny Chen came up with some beautiful solutions for this. So we have an impinging jet flow, which is very stable and very tolerant to changes in design and changes in breathing pattern. With a Peltier chip condenser, temperature sensors on the inlet and outlet, and flow measurement so that we could correct for breathing patterns, and this allows the patients to breathe normally, which is a really important part for COPD patients. So this is the competing technology, the R tubes, and we see the patient blows in here. It goes round the bend. There'll be all kinds of helical variable flow here. It condenses because this stainless steel sleeve has been in the freezer. And I don't know about you, but do you really know what temperature your freezer is? Really? How long has it been since been defrosted? How long has the stainless steel sleeve been on the side? There's no way this could be getting reliable results. And this is our device, since commercialized by Danny Chen and Xiao Kai Hong, two of our great students. And they set up a company called Respire Diagnostics, so you can now buy this. You blow into this, it condenses on the Peltier chip at the back. We've got all the sensors to measure all this stuff. We get around 30 to 50 microliters in 2 to 5 minutes of normal breathing, which is similar to the R-tube technology, except it works better and is properly controlled. And we can keep the samples. We've got a hydrogen peroxide sensor based on Prussian blue, so we've coupled the redox chemistry of iron 2 and iron 3 to the hydrogen peroxide chemistry here. We have to look at all the different steps in this and back out all of the rates of these reactions. And we get limits of detection down in the sub-micromolar level and repeatable measurements this way. And we can also measure nitrite as well at the same time. So by combining hydrogen peroxide with nitrite and the flow data, because the pattern of velocity in patients with COPD is different from normal healthy individuals. And combining these into a machine learning classifier, it turns out to be as potential as a diagnostic, which we're very surprised about as well, because we're really just looking at a healthcare device that allowing people to manage their own healthcare conditions. And we can couple this to an enzyme to measure lactate in exhale breath as well. These are the raw data for those of you who think I'm just trying to hide through this stuff. And these are the disposable sensors that we're manufacturing at scale now. And we did some preliminary evaluation in healthy exercise volunteers. Too damned healthy in the case of Richard Wilson. We had to get him up to 210 watts before he produced any lactate at all. So don't use healthy individuals if you're trying to study lactate. Ignoring the first point for a moment, see when he exercises, the lactate on his breath goes up and goes down when he stops cycling. We've got this intriguing business at the beginning there that really made me wonder, were we measuring something else as well? So we developed an LC-MS assay with Alain Ziek in infectious diseases, did some fantastic analytical chemistry for us, and no, all of these results that our sensor got were all actually correct. So this early stage fall in lactate when patients healthy volunteers are getting ready to exercise, we have no idea about. We've tried all sorts. We saw there's bugs on teeth, what you've been eating, someone having a sneaky bottle of Duchesse de Bourgogne, which has got lactic acid in it beforehand. None of these are an explanation. But the results all fall in line with the mass spec, and we can't argue with that. So, in conclusion, if you're going to get into diagnostics and bioanalysis, you need interdisciplinary knowledge. So you need to know what you don't know, and then you hire the experts to deal with this stuff. You build a multidisciplinary team, relying on your own sketchy knowledge to actually identify people who have the core knowledge that can make your project work. And this has been a central element to my research throughout my professional life. I think one of my major conclusions is the ideas are the easy bit, and the reduction to practice is where things go horribly wrong, and where it gets hard. Minimally invasive technology can address some of these demographic challenges in healthcare deliveries, and more importantly, get better patient outcomes. Interstitial fluid is an accessible medium, low risk. We can measure all sorts in with the microneedles. An exhale breath condensate we think is a viable potential source. But really, my final conclusion, valid analytical measurements are difficult to do. Lots of people paid for all of this. This is my favorite quote from one of my favorite books, Gulliver's Travels by Swift. He meets the scientists on the flying island, and they take him back to their homeland where their labs are. And he's describing the extraction of sunbeams from cucumbers. And he said, you know, young students were employed in this labor, which might still be improved and much expedited if the public would raise a fund. And thank you all for your attention. You've been a wonderful audience. I'm all done.
I'm not going to say thank you and congratulations. That will come later. But this was absolutely fantastic. We have time for just a few questions from either the audience in the lecture theatre here or online. Shulin is scanning the online chat on YouTube to see if anybody wants to make a question. They're 20 seconds behind us, so that gives us time.
That was in case I swore, right?
We weren't counting, Danny. Is there a question here in the room? Molly.
So, Danny was amazing and very, very entertaining.
Thank you, Molly.
For the microneedle sensing, for the microneedle sensing, you could do so many other applications as well, right? Where are you thinking for the future?
Well, the host factors for infection, and what we want to be able to do is titrate antibiotics against the patient's own response to an infection. So if we could measure those either going down as the treatment's being successful or not changing as a result of the antibiotic, and you know they're resistant, I think that's a very exciting application. And the same idea could be applied to other drugs. And we're actually applying the same concept to not to microneedles, but to urine samples for urinary tract infections as well. So you try and measure the drug and the production of nitrite from nitrate by the bugs. So that's definitely an area I'm super excited about. I think there are all kinds of other areas that would occur to anyone else, like drug testing, for example, or Bale conditions, stuff I'm not interested in.
Well, I think if you can make a dent in the antimicrobial resistance, it's hugely important.
I mean, it's probably third most serious existential threat to humanity at the moment. I think maybe something like that. So it definitely seems like a worthwhile activity. Yeah.
Thanks.
Thanks, Mike.
Any other questions from in the room? I know that we have refreshments afterwards, so you'll have time to collar Danny and ask him those questions that you don't want to raise in public.
I can see about 100 people here dying of thirst.
There's
Frank.
one here. Here we go.
With regards to the condensation using Peltier, is there also not an informed amount of condensation effects of the aerosol that might be forming when the breath is first released in between that and the Peltier that would be critical
Frank, were you sitting in on the discussion that Shu Lin and I were having the other day? Because when we calculate the levels of lactate that we should find, they're 5 orders of magnitude lower than the ones we actually get. So it's definitely aerosols. Absolutely aerosols. And we don't know what this means. And we're absolutely open if there's anybody on YouTube who's got an idea, respiratory physicians in particular. And we've been talking to people, we've been constantly talking to respiratory physicians about how to analyze this. If we knew where they were coming from, it'd be easier to interpret. But no, absolutely right. It is, we think, mostly aerosols that we're capturing. Because one tiny droplet of actual liquid is many, many more times more molecules than a vapor. So yeah, absolutely. We should get you back in the lab. Now you're retired.
We're going to take our final question from online. Shu Lin, do you want to read it out?
Yeah, so here's one online question. In the area of AI, how much do we need accurate biomeasurements vs a large number of them?
Oh yeah, I mean, I think this is going to be an important field. And I would really suggest they talk to somebody who knows about AI about this, because it's absolutely not my specialty. But I think the idea of just looking for patterns and not caring what they are is definitely an important trend in diagnostics. But it's not something that I would pretend to have any expertise in here. There are, mercifully, however, plenty of people in the Department of Bioengineering at Imperial College who do, and I would encourage them to get in touch with, for example, Anil Barath.
Danny, if I could invite you just to take a seat for
Absolutely.
a moment and invite Peter Winlow to come up. Peter is Professor of Biophysics at Exeter, a graduate of Imperial College Physics, and was here in bioengineering, well, before it was called bioengineering, for a couple of decades, over 20 years, I think. And you've already seen his photo today. This is what he looks like in the flesh. Actually, no different, I would say. And he clearly overlapped substantially with Danny in his time here. And it's a great pleasure that Peter is able to be here to give the vote of thanks.
Right, well, I'm sure you all want to join me in saying thank goodness that's over. And that Danny at long last has got the chair that he deserved for so many years. But I'm sure he didn't invite me here to say nice things about him. So I thought I might take you back to the beginning, to where it all began. Now, round about 1980, Kim Parker, my oldest and dearest friend, who I'm delighted to see here this afternoon, were trying to carry on the work of a Polish postdoc who we're convinced was a spy and just doing a little research on it. But he left us with the problem of measuring oxygen concentrations in the walls of blood vessels. And Kim set about assembling our very first computer. And after several long weekends, and with the aid of a reel-to-reel tape recorder as the data acquisition system, we were able to record electrode currents of the order of 1,000 data points in no more than 10 seconds or so. So this revolutionized chronoamperometry. We realized that, but we also realized we didn't know any electrochemistry. But we, as Danny pointed out, realized that one of the world's leading electrochemists was sitting over in chemistry. So we beat a path to his door. And he was very, very understanding. He entertained us for many afternoons and really took us under his wing. And then finally, he introduced us to this young chap who was skulking about in his laboratory, having screwed up his final examinations. And I often wonder whether that was a joke on his part, because this electrochemist was a man of Catholic taste in the widest sense of the word. And I always suspected this was because he couldn't persuade Danny to join him in some of his more priestly behaviors. Anyway, we had to make a special appeal to some college board or other to allow Danny to undertake his PhD. And despite all of that, it worked out very well. Danny very quickly mastered all the electrochemistry we threw at him. The one thing he found difficult was that because we work at the interface between physical, biological science and medicine, it means you have to interact with people from very different backgrounds and work in different people's labs. And that was something that Danny struggled with for a long, long time. One of my first experiences was going with him to to one of the biological facilities here, and we were given a lab where, for some reason, they had a large poster on the wall on how to anaesthetize a pig. And it was a very graphic poster, and it led Danny and I to a very animated discussion about animal husbandry, which we thought was very far-reaching and extremely entertaining. But unfortunately, the technicians in the lab didn't share our sense of humour. And strong letters were exchanged between heads of department, which resulted in us both being banned from the lab for some considerable time. In fact, I'm not sure whether that ban has actually been... So that was a very good start. And Danny's first project concerned measuring oxygen concentrations in the intervertebral disc. And so it was inevitable that I brought him into contact with the queen of the intervertebral disc, my other friend, who was incredibly bright, but also very reserved and doesn't tolerate fools gladly. So every student is in awe of her. And so I put it off as long as possible, but before the end of his PhD, he and I had to make a trip up to Oxford to put his electrodes into an intervertebral disc preparation of hers. And we had a great day, but I got very nervous when I realized that I had to leave and drive back to London, leaving he and this lady alone together. And so the next morning, I got on the phone to see how things went. And I found out that she was even frostier than usual. And it took me a long while to thaw her out. And it turned out that Danny had assured her that he was perfectly equipped not only to solve outstanding problems in the intervertebral disc, but also to resolve any inadequacies she may be finding in the marital bed. I got no idea at all how that conversation evolved. Anyway, there are many more stories I can tell you about Danny, but most of them would be slightly tasteless, so I think they're probably better left till later on. So I shall finish with the observation that when Danny went into exile in Brighton, I thought he really belonged here and tried very, very hard to find ways to bring him back. They all failed, and the only way I could find of bringing him back was to leave myself. And I'm sure Anthony and other people in the department will tell you that that's a win-win situation. But before I give him that opportunity, I think again I'll stop talking and just ask you to join me in thanking Danny for a great presentation and congratulating him on his position. Very well done.
I thought he was going to complete the pig story.
Okay, everyone. Let's celebrate with Danny.
Yay. Go and have a drink.